Hello students, my name is Niyati Said and thanks for watching Edupedia word videos. My topic for the presentation is ninth section of the chapter Mineral Nutrition. In this section of the video, we will discuss about metabolism of nitrogen students. Okay, so metabolism of nitrogen or nitrogen cycle. You may have learned about the water cycle or carbon cycle before. These cycles move substance, water or carbon throughout the environment and organism living there. The process is fairly simple, students. However, the movement of nitrogen through living and non-living things is a bit more complex. The nitrogen cycle is more complex than other biogeochemical cycle, students. Please note that this is the most complex cycle than other biogeochemical cycles because most organisms cannot use the nitrogen found in the atmosphere, even though almost 80% of the air is nitrogen, students. In the atmosphere, nitrogen is found as N2. Okay, this N2 is not usable by most plants and animals. In order for plants and humans to get the nitrogen they need, the nitrogen must be converted into ammonia form. Okay, that is NH3. So the conversion of this atmospheric nitrogen to usable form, that is ammonia, this is known as nitrogen fixation. Okay, and the nitrogen fixation can be defined as the process of creating ammonia from atmospheric nitrogen. Without this process, students, most plants and animals would not have nitrogen needed to live. Okay, as you can see that this is the atmospheric nitrogen which is taken up by the plant. See, plant uptake in the form of nitrate. Okay, see, this is taken up by the plants in the form of nitrate, and then nitrification takes place. Okay, or you can say that see, this is the cow and it has given the compost in the form of organic matter. Okay, this is your ammonia which gets converted into NO2 minus, and this plants, they uptake nitrate or nitrogen in the form of nitrate. Okay, so nitrogen is the most prevalent element in living organism, students. Okay, plants compete with microbes. Okay, as I told you that plants, they compete with microbes for limited nitrogen in soil. Thus, nitrogen is limiting nutrient for natural and agriculture ecosystem. The process of conversion of nitrogen, that is N2 to ammonia, is called as nitrogen fixation, students. Okay, lightning and UV radiation, they provide energy to convert nitrogen to nitrogen oxide, that is NO, NO2 and N2O. Okay, see, this is a lightning and UV radiation which provides energy to convert N2 into NO or NO2 or N2O. Okay, so industrial combustion, forest fires, automobile exhaust and power generating stations are also source of atmospheric nitrogen oxides, students. Okay, now question arises, what's the significance of nitrogen to plants? Now that we know that nitrogen fixing bacteria, they convert atmospheric nitrogen into ammonia, we need to look at why this matters. So what if plants can't use the nitrogen in the air? What do they really need nitrogen for anyway? We know that plants, they need elements such as carbon, hydrogen, oxygen for photosynthesis, but nitrogen is actually also needed for this process as well. Chlorophyll, the green pigment found in the chloroplast of plant cell, is responsible for photosynthesis. We very well know that. One of the element that helps make chlorophyll is nitrogen. Without nitrogen, plant would not be able to perform photosynthesis, students. Okay, plants, they also need nitrogen because it is found in all living cells. Having the proper amount of nitrogen, it helps with plant grow. and plants that have right amount of nitrogen will have more seed and fruit production. Okay, so this nutrient, that is nitrogen, improves the quality of leaf also. If plants do not have enough nitrogen, they will not be able to function properly, because of this nitrogen is found in many fertilizers. Okay, see, this lightning is producing the energy to convert nitrogen into usable form. So nitrogen fixing bacteria are present in the roots of the leguminous plants, so they trap nitrogen and convert into the usable form. Okay, see this nitrogen fixing bacteria in the soil, it gets converted, nitrogen is converted into nitrate, and thus it is absorbed by the plant. Okay, I'll tell you what is denitrifying bacteria, but here just know that how this nitrogen gets converted into the usable form, that is nitrate. Okay, nitrogen is the most prevalent element in living organism. We very well know that apart from carbon, hydrogen and oxygen, it is the most prevalent element in the living organism. And nitrogen is a constituent of amino acid, proteins, hormones, chlorophyll and many of the vitamins. Plant, they compete with microbes for limited nitrogen that is available in soil, students. Thus nitrogen is a limiting nutrient for both natural and agricultural ecosystem, students. Okay, the process of conversion of nitrogen to ammonia is termed as nitrogen fixation. Lightning, as you can see, this provides energy to convert nitrogen to nitrogen oxides. Okay, industrial combustions, forest fires, automobile exhaust and power generating stations are also the source of atmospheric nitrogen oxides. Please note that. Okay, so this is the nitrogen cycle. Okay, see, nitrogen, it gets into the plant protein and from dead organic matter. Okay, and then it gets converted into the ammonia gets converted into nitrite ion. That process is known as nitrification, and then it gets converted into nitrate, or you can say that this is how nutrient uptake takes place. Okay, nitrogen mineralization, decomposition and oxidation by cellulomonas bacteria, and then nitrification takes place under the nitrosomonas bacteria, and NO2 minus gets converted into NO3 minus, and this takes under nitrosomonas bacteria. So what's the process in the nitrogen cycle fixation, students? Fixation is the first step in the process of making nitrogen usable by plants. Here bacteria which is present in the root of the leguminous plants, they change nitrogen into ammonium form, students. Okay, so what is nitrification? This is the process by which ammonium gets changed into nitrates, that is NO3 minus, by bacteria. Nitrates are what the plants can then absorb. See, plants cannot absorb nitrogen as such. So when it gets converted into nitrate form, then plant can absorb it. Okay. Now comes the assimilation part. Assimilation, this is how plant get nitrogen. They absorb nitrate from the soil into their roots. Then nitrogen get used in amino acids, okay, or nucleic acids and chlorophyll. Then comes ammonification. Ammonification, this is the part of decaying process. When a plant or animal dies, decomposers like fungi and bacteria, they turn nitrogen back into ammonia. Okay, so it can re-enter the nitrogen cycle. This is as simple as that. And then denitrification, students. Extra nitrogen in the soil gets put back out into the air. There is a special bacteria that perform this task as well. Okay, I'll explain you in detail what it is, but for the time being, just know that these are the 4 steps in the fixation of nitrogen. First is nitrification, where ammonia gets changed into nitrate, that is NO3 minus, by bacteria. And then assimilation, this is how plant get nitrogen. They absorb nitrate from soil into their root, and then nitrogen gets used in amino acid, nucleic acids, and chlorophyll, as I told you. Okay, that nitrogen is a constituent of amino acids, protein, hormones, chlorophyll, and many of the vitamins. Okay, and then comes ammonification. This is the part of decaying process. When a plant or animal dies, decomposes like fungi and bacteria, they turn nitrogen back into ammonia, and so that it can re-enter the nitrogen cycle again. Then comes denitrification. Extra nitrogen in the soil gets put back out into the air. Okay, and there is a special bacteria which performs this task very well, and those bacteria are known as pseudomonas and thiobacillus. Okay, now see, as I told you that atmospheric nitrogen can be fixed in three ways, that is biological nitrogen fixation, industrial nitrogen fixation, and electrical nitrogen fixation, students. Okay, so biological nitrogen fixation means very few living organism can utilize the nitrogen in the form of N2 available abundantly in the air. Only certain prokaryotic species are capable of fixing nitrogen. Reduction of nitrogen to ammonia by living organism is known as biological nitrogen fixation. The enzyme nitrogenase is capable of nitrogen reduction, okay, which is present exclusively in prokaryotes and not in eukaryotes. Okay, such microbes are called as nitrogen fixers. Okay, then comes industrial nitrogen fixation. This also converts nitrogen into ammonia, that is NH3, and electrical nitrogen fixation, that is your UV radiation, and that is your electrical nitrogen fixation. So it converts N2 into NO3 form and not in the ammonia form. Okay, so biological nitrogen fixation and industrial nitrogen fixation, it converts N2 into NH3, that is ammonia, and then it gets converted into NO2 minus, that is nitrite ion, and then eventually it gets converted into nitrate ion. So electrical nitrogen fixation and denitrification, it converts into nitrate form. Okay, nitrate is uptaken by the plant biomass, and then when it dies, or when plant or any animal dies, it or decays, then fungi and some bacteria, it helps in the decaying process, and then it fixes or it converts the nitrogen present in the plant and animal body into the ammonia form, so that it can re-enter the nitrogen cycle again. Okay, so decomposition of organic nitrogen of dead plant and animals into ammonia is called as ammonification. So this is your ammonification. Okay, some of this ammonia volatilizes and re-enters the atmosphere, but most of it is oxidized into nitrate by soil nitrifying bacteria, that is Nitrosomonas, Nitrococcus, and Nitrobacter. These are your chemoautotrophs. Okay, these nitrifying bacteria are called as chemoautotrophs. See, 2NH3 plus 3O2 in the presence of Nitrosomonas bacteria and Nitrococcus bacteria, it gets converted into nitrite ion, that is NO2 minus, or hydrogen is liberated and water is liberated. Okay, now this NO2 here, it becomes reactant in the presence of oxygen, in the presence of Nitrobacter, that is your nitrifying bacteria, which fixes the nitrogen and it gets converted into NO3 minus, that is your nitrate ion, students. Okay, these steps are called as nitrification. Ammonia is first oxidized to nitrite. Okay, see, as you can see that this ammonia is oxidized into nitrite by the bacteria Nitrosomonas and Nitrococcus. The nitrite is further oxidized in the presence of Nitrobacter to nitrate with the help of bacterium Nitrobacter. Okay, so these steps are called as nitrification. These nitrifying bacteria are called as chemoautotrophs. Okay. Plants, they absorb nitrate and is transported to the leaf. Okay, in leaves it is reduced to form ammonia that finally forms the amine group of amino acid, because nitrogen is an important constituent of amino acid proteins. Okay, so nitrate present in the soil is also reduced to nitrogen by the process of denitrification. As I told you that denitrification is carried out by bacteria Pseudomonas and Thiobacillus. Denitrification, it converts nitrogen present in the decayed animal or plant body, and it converts into the ammonia so that it can re-enter the nitrogen cycle. Okay, and can be utilized. So plant fixing bacteria, see, this is nitrogen which is present in the air, and these are the root nodules which are present in the pulse crop or in the leguminous crops. So these microbes, they reside in those root nodules and they fix nitrogen in the nodule on the root of the pulse crop, students. Okay, so it is the reduction of nitrogen to NH3 by living organism in presence of nitrogenase enzyme, students. See, conversion of nitrogen, N triple bond N, it is converted into NH3 in the presence of nitrogenase enzyme. Okay, and very few, as I told you, that very few living organism can utilize nitrogen in the form of nitrogen, which is available abundantly in the air. Only certain prokaryotic species are capable of fixing nitrogen. Reduction of nitrogen to ammonia by living organism is called as biological nitrogen fixation. Okay, and those who fix nitrogen, they are called as nitrogen fixers. Okay, students, please note that nitrogen fixing microbes could be free living or symbiotic. Examples of free living nitrogen fixing aerobic microbes are Agitobacter, okay, while Rhodospirillum is anaerobic and Bacillus is free living. Okay, so in addition, a number of cyanobacteria such as Anabina and Nostoc are also free living nitrogen fixers. See, as I told you that nitrogen fixing microbes are of two types. First is free living. Examples are Agitobacter and Bizarnica. These are aerobic microbes, and Rhodospirillum and Bacillus, they are anaerobic, and cyanobacteria such as Anabina and Nostoc. Okay, and now comes symbiotic. So symbiotic are Rhizobium. Okay, now these are the organisms and this is the status of these organisms. Clostridium is non-photosynthetic bacteria. It is anaerobic. Okay, that means it doesn't require oxygen to carry out its function. Then Klebsiella is also non-photosynthetic and it is facultative bacteria. And Agitobacter is also non-photosynthetic and it is aerobic bacteria. Then comes Rhodospirillum, which is purple in color and it is non-sulfur bacteria. It is photosynthetic. And Anabina is photosynthetic bacteria and it status of Anabina is cyanobacteria. Okay, okay students. Symbiotic biological nitrogen fixation, what it is? See, this is the leguminous plant or any pulse plant. Okay, so it has root nodules or root knots present in the root where microbes reside, where they fix nitrogen into the usable form. Okay, so this is the zoom view of these microbes which is residing in the root nodules of leguminous plant. This is the rhizobium which is present in the nodule, and this is the leg hemoglobin which is responsible for fixing nitrogen and converting it into the usable form of nitrogen, students. So legume bacteria relationship most prominent. Okay, for example, rhizobium bacteria, these are the rod shaped bacteria. This is rhizobium bacteria, it is rod shape as you can see in root of the Legumes. Legumes plants are such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover bean. These are your um leguminous plants. Okay. The most common association on root is as nodules. The microfrankia also produces nitrogen fixing nodules on the root of non-leguminous plants. For example, alnus. Rhizobium and Frankia, they are free living in soil, but as symbionts can fix atmospheric nitrogen. Okay, central part of the nodule is red or pink color, as you can see, due to leguminous hemoglobin, which is called as leg hemoglobin. Okay, leg means leguminous and hemoglobin, so leg plus hemoglobin is equals to leg hemoglobin. So this is the central part of the nodule, which is pink or red color due to the presence of leguminous hemoglobin. Okay. So what are the principal stages in the nodule formation, students? See, this is the rhizobia. Okay, multiply and they colonize the surrounding of the root, and they get attached to the epidermal and root hair cells. See, these are the root hair cells and these are the soil particles, and these are the bacteria. And bacteria, it goes closer to the root hairs, and thus it enters the plant body by inner cortex and pericycle cells under division. So this way they infect plant body, and thus forms a mature nodule. Okay. So, nodule formation involves a sequence of multiple interaction between rhizobium and root of the host plant. There are principal stages in the nodule formation. First, rhizobia, it multiply. As you can see, rhizobia is a bacteria. So these are the bacteria. Okay. So rhizobia, they multiply and they colonized the surrounding of root, and they get attached to the epidermal and root hair cells. See, this way it gets attached to the root hair cells. The root hairs then curl. As you can see, that root hair has gone curled, and bacteria, this way it invades the root hair. An infection thread is produced carrying bacteria into the cortex of the root. See, in this way it enters the cortex of the root, where they initiate nodule formation in the cortex of the root. Then the bacteria are released from the thread into the cell, which leads to the differentiation of specialized nitrogen fixing cells. Okay. The nodule thus formed establishes a direct vascular connection with the host for exchange of nutrients. So this is a symbiotic you can say symbiotic association. Okay. So a nodule, see, this is a mature nodule which contains all necessary biochemical composition or components such as enzyme nitrogenase and leg hemoglobin. The enzyme nitrogenase is a molybdenum iron protein, and it catalyzes the conversion of atmospheric nitrogen to ammonia. I'll explain you in detail. So this is how a nodule, it establishes a direct vascular connection with the host for exchange of nutrient, because it forms a symbiotic association, students. Okay. So this is how it is. Steps of the conversion of atmospheric nitrogen to ammonia by nitrogenase enzyme, which is found in nitrogen fixing bacteria. See, this is enzyme nitrogenase, and substrate of this enzyme is nitrogen. So N2, that is nitrogen, atmospheric nitrogen. So it gets fit into this substrate, that is your enzyme, and then it gets reduced. That means hydrogen gets attached to this nitrogen molecules, and then again two molecules of hydrogen gets attached. That means again reduction takes place, and then again reduction takes place of again two molecules of hydrogen, and this way it produces NH3. If you break this, so two molecules of NH3 are evolved. So release of the product takes place, and free nitrogenase this is free nitrogenase, okay. As you can see, this is enzyme nitrogenase, so it can bind another molecule of N2. So this way you can say it fixes nitrogen, okay. So the nodule contains nitrogenase enzyme and leg hemoglobin. Nitrogenase, as I told you, that it is MOFE protein, that is molybdenum iron protein, and it catalyzes the conversion of atmospheric nitrogen to ammonia, okay, which is a first stable product of nitrogen fixation. Ammonia synthesis needs high input energy, that means it requires 8 ATP for each ammonia formation. It is obtained from the respiration of host cell. So this is the reaction which takes place in the biological nitrogen fixation. That means when N2 gets fixed, then it requires 16 ATP to fix two molecules of ammonia, and thus it liberates ATP, gets converted into ADP and inorganic phosphate. That PI is inorganic phosphate, students, okay. So this is the leg hemoglobin, which is oxygen scavenger, okay. It is red or pinkish portion, central portion. So what makes the nodule pink? This is due to the presence of the leguminous hemoglobin or leg hemoglobin, okay. So nitrogenase enzyme is highly sensitive to molecular oxygen. It requires anaerobic condition, okay, to protect from oxygen. For this, leg hemoglobin act as oxygen scavenger. That means it captures it, okay. So these microbes, they live as aerobes under free living condition where nitrogenase is not operational, but during nitrogen fixing event they become anaerobic so that they can protect nitrogenase enzyme, students, okay. Now comes reductive amination and transamination, okay. So what it is, students? Fate of ammonia. At physiological pH, ammonia is protonated to form ammonium ion, okay. While most of the plants can assimilate nitrate as well as ammonium ion, the latter is quite toxic to plants and hence cannot accumulate in them, okay. Let us now see how ammonium ion is used to synthesize amino acid in plants. There are two main ways in which they can take place. So first is reductive amination and second is transamination, students. So first is your reductive amination. Reductive amination, in this process, ammonia reacts with alpha ketoglutarate acid, okay. Ammonia react with alpha ketoglutarate acid to form glutamic acid, students, as indicated in this equation that is given below. So alpha ketoglutarate acid in presence of ammonium ion and NADPH, it gets converted into glutamate or water and NADPH, okay. So this is how it is. And then comes your transamination, students. What is transamination? It involves transfer of amino group from one amino acid to the keto group of keto acid. So it is a transfer of amino group. That's why it is called as transamination, because transfer of amino group takes place from one amino acid to the keto group of the keto acid in presence of transaminase enzyme, okay. So glutamic acid is the main amino acid from which transfer of NH2 takes place and other amino acids are formed through transamination, okay. See, this is the amino donor, this is one amino acid and this is another amino acid. So this is amino acid donor and this is amino acid acceptor. That's why transfer of amino group takes place between these two amino donor and amino acceptor. That's why this is known as transamination, okay. So this is how it is. That means, see, R1CO, this CO comes here in this place, and this goes into this place. See, CH and H3 plus. Okay, so glutamic acid is the main amino acid from which transfer of NH2 takes place, and other amino acids are formed through transamination. Okay, so asparagine and glutamic acids are the two most important amides which is found in plant as a structural part of protein. They are formed from two amino acids, namely aspartic acid and glutamic acid, respectively, by addition of another amino group to each. Okay, so the hydroxyl part of the acid is replaced by another NH2 minus radical, as I have shown you. Since amides they contain more nitrogen than amino acid, they are transported to other parts of the plant via xylem vessel. Xylem is a conductive tissue. So in addition, along with the transpiration stream, nodule of such plant, that is soybean, okay, they export fixed nitrogen as ureas. These compounds also have particularly high nitrogen to carbon ratio. Okay, so this is how fate of ammonia decides. There are two main ways by which this can take place. First is reductive amination, which I have taught you, and second is transamination, which I just taught you. Okay, so this was all about metabolism of nitrogen, and this ends the chapter mineral nutrition. Thank you.